All right, we're going to start our next section. We're going to start off talking about post-traumatic stress disorder, and then on Wednesday we have 3 papers that we're going to discuss, relatively easy papers that we'll talk about with PTSD, and we'll try to fit case studies in somewhere along the way. We're still kind of playing catch up from being out that week. So, we're talking about the stress system today with post-traumatic stress disorder. So PTSD, it's a condition that occurs when somebody has experienced a near-death experience or had their life threatened or seen other traumatic events happen, or people that have been in an abusive relationship is another thing that can cause PTSD. And we'll talk about the symptoms, we'll talk about the diagnosis on the next slide, but the symptoms include particularly having debilitating recollections of the event, right? And that can happen in sleep. We'll talk about the role of sleep in this. It can happen while they're awake as well. Often those debilitating recollections are triggered by environmental stimuli that are associated with the PTSD. There's a pretty wide sort of range of prevalence. Right now, I think the latest figures were around 4% prevalence in the general United States population. But in subpopulations, that's very different. Right? And in particular, populations where people would maybe frequently see traumatic events. And so that can be in first responders is a big subpopulation of people that are susceptible to PTSD, and people that have seen combat. So U.S. combat veterans have a very high rate of PTSD, about 17%, compared to 4% in the general sort of population. And we have pretty good data on this because of well, the fact that the military keeps track of this, and when people have PTSD and they're having issues doing their job in the military, they get logged as such, right? We don't have as good numbers on things like first responders. We don't really know as well how many first responders are affected by PTSD. This 17%, about 23% of veterans that seek aid at a VA hospital after they have returned back to the States are showing PTSD. And so an even higher number of people are actually seen in the VA hospital system. So the DSM-5 criterion, there's a bunch, right? So first, pull all this up. So exposure to actual or threatened death, serious injury, or sexual violence. A couple of ways listed there. And then, so this is the thing that causes, right, the etiology of PTSD, right? But then the presence of intrusive symptoms, the presence of persistent avoidance of stimuli that may be triggers of the PTSD, alterations in cognitive thought patterns and mood, a bunch of 7 different criteria under that, and then changes in arousal and reactivity, exaggerated startle response, problems with concentration, hypervigilance is a big one that we'll see in some of the case studies. And this has to last more than a month, right? So, you know, you're in the military with your platoon and you get hit by a rocket and people die in your motor brigade or whatever, and obviously that's going to be traumatic, right? Everybody's going to have some trauma associated with that. But if it persists more than a month and if it's debilitating, right, are the 2 sort of criteria of where it goes from being normal trauma response to abnormal, to where it's diagnosed. All right. And then there's some other criteria about dissociative symptoms, you don't have to really worry about that. All right, so to summarize all that, so that's the full DSM-5 criterion with all the little intricacies of it. But basically, the thing you need to know is exposure to actual threat and death, serious injury, or sexual violence, intrusive symptoms, avoidance of stimuli, changes in cognition and mood, and changes in arousal and reactivity. Again, more than a month, and that it's clinically impairing their ability to function. All right, so how does this happen, right? Well, to talk about PTSD, we got to talk about the stress system first and really dive in and understand what the stress system is doing in our body. So our stress system consists of the HPA axis. I've drawn that out over here on the board. We'll talk a little bit more about this in a second. But we start out with the hypothalamus, in particular the PVN, the paraventricular nucleus of the hypothalamus. And that then is going to release a signal called corticotrophin releasing factor. It's also sometimes abbreviated, it's abbreviated CRF, corticotrophin releasing factor, or CRH, corticotrophin releasing hormone. Same thing, interchangeable words, okay, here. And that activates part of the anterior pituitary. The anterior pituitary releases ACTH, this hormone, that then activates the adrenal gland, and the adrenal gland is then going to release cortisol or glucocorticoids. And I'll use glucocorticoids and cortisol sort of interchangeably. It's the same name for both things. It's one chemical. And the stress, so why do we have this cascade effect? Well, we have a very small number of neurons in the hypothalamus, right, that can communicate to a dedicated group of cells in the anterior pituitary to release ACTH. Then the ACTH goes to this big endocrine gland down here, the adrenal cortex, that is designed to pump out tons of cortisol, right? And so we have this cascading factor to magnify that effect so that we have a dedicated organ that's just releasing cortisol. And there are glucocorticoid receptors on almost every cell in the body, so stress affects all of the processes in our body, including in our brain as well. All right. Now, we have a short term, so what is the, let me ask this, what is the point of our stress system? Why do we have it? I know it's Monday, but we can talk. It's all right. You had a whole day break to rest up. I'm sure you thought the whole time, everybody's going to ask me questions on it. Why do we have a stress system?
Survival mechanism.
A survival mechanism, right? Do animals have stress systems? What do they use their stress systems for? To escape predators. Very good. I'll find your voice. All right. Yeah, so our fight or flight system, right, is a survival system. And so we have two kind of systems. We have a short-term immediate system. That's our fight or flight, right? And that's going to be the locus coeruleus, short-term, releasing norepinephrine to trigger all these things, to cause arousal in the brain and trigger all of this. And we have a dedicated part of the adrenal gland. So the adrenal gland has the medulla, which is an internal part, and the cortex, which is the surrounding part, just kind of like the brain has a medulla and a cortex, right? The medulla is part of the sympathetic nervous system and helps trigger releasing norepinephrine throughout our body. So in our brain, locus coeruleus releases norepinephrine. In our body, the adrenal medulla releases norepinephrine. And that's our immediate, like, run, energize, but that's very short. That's very short-lived. It's like kick-starting the system. And then this happens, and cortisol takes over, and cortisol is sort of our sustained upregulation in energy. And so cortisol is going to do the same sorts of things that norepinephrine, our sympathetic nervous system, does. It's going to shut down our digestion. We don't need to be digesting food if we're about to be eaten by a lion, right? We need to run and get away, right? It's going to turn on our immune system, It's going to upregulate our immune system because if we get cut or bit by the lion, right, we want to have our immune system in full strength. It's going to divert all of our energy away from things like digestion and other sort of prolonged survival things into our muscles, into our escape systems. Right? And it's going to stimulate our hippocampus. What's the hippocampus involved with? Memory formation, right? So we want to remember all the events that led up to the lion chasing us through the savannah if I'm a zebra, right? Because if I see or hear any of those cues or smell any of those cues again in the future, I want to get a jump start on running, right? So I want to have memory triggered in response to this stressor so that I can escape. How long does the normal stress response last? What's your guess in an animal? So an animal, zebra, going to get water down the water hole and a lion jumps out. Stress response runs away. How long do you think that lasts? Take a guess. Half an hour, about 15, 20 minutes, yeah. Right? Long enough to get away. And then we got to turn everything down because we're expending a ton of energy. Right? This is using a ton of energy. This whole system is designed to shut itself down. So it has what we call a negative feedback loop. Negative feedback. It's designed to turn itself off through these GR glucocorticoid receptors. We have two different types of receptors, we'll talk about this in a minute. MR and GR, both of them are stimulated by glucocorticoids. It's designed to shut itself down so when the physical stressor is gone, we can return to homeostasis. Right? We can return to homeostasis, we can quit diverting energy. Right? The problem is that unlike zebras, right? Zebras aren't walking around the African savannah thinking, is there a line over there? Is there a line over there? Is there a line over there? No. But cognitively, they're not thinking about that. They are reacting to stimuli, reacting to the presence of a physical threat, a physical stressor. However, as humans, we create this lovely brain of ours that has psychological stressors. We do walk around thinking about things that might happen in the future, right? And that can generate stress, right? Am I going to do well in this class? That can generate stress. And psychological stressors override this negative feedback loop, so they can cause the system to continue to pump out cortisol, continue to divert energy, and that becomes a very negative state. All right. So as I mentioned, we have two different types of receptors. Mineralocorticoid, that's the MR receptors. Glucocorticoid, those are the GR receptors. The GR receptors are more widely found in the brain. The mineralocorticoid are more for that sort of memory formation. They're primarily located in the hippocampus. And prolonged exposure can cause excitotoxicity that we've talked about, right? So if we stimulate these areas too much for too long, we can overwhelm the system. Too much calcium comes in, can cause excitotoxicity. The amygdala is also involved. It has GR receptors, right? The amygdala involved with our emotions. So we're going to assign very negative emotions to all the things that are happening when we're under stress, because we want to, again, the whole point of this system is to remember and avoid that in the future, right? It was designed to deal with physical threats, not with psychological threats, right? Psychological stressors. And then, as I mentioned, the locus releases, that's a short-term system in our brain to cause hypervigilance. All right. So again, if we look at the distribution of these, the MR, this is in a rat brain, so here's our hippocampus, primarily found in the hippocampus. GR receptors also found in the hippocampus, but also, right, a high density of glucocorticoid receptors in the PDN. What are they doing? What's the job of these GR receptors in the tibia? What are they doing over here? Shutting down the system. That's our negative feedback system, right? This is when cortisol binds these receptors, it says don't release more corticotrophin releasing factor. Turn off the system, right? Again, high density in the amygdala here, right? So these are all really important players, right? Hypothalamus, PDN is turning it off. Hippocampus is forming memories. Amygdala is assigning negative emotional value. All these things are really important. All right. So short-term stressors or physical stressors enhances memory formation that we talked about. Although sometimes the accuracy isn't great on that. So it's not always particularly accurate, but we do form memories associated with events leading up to that. Cahill and McGall did a famous study at UC Irvine where they had two stories. One story was about a boy and his mother who went out shopping for the day and talks about going into stores, buying some gifts, and that story had low emotional impact. The second story was about a boy and his mother going out shopping, except the boy gets hit by a car crossing the street and is rushed to the emergency room and is in serious condition, and that one generates more emotional content. Right? When the next day, when they brought the subjects back in, or a week later, or whatever, some delay, they bring them back in and say, tell us details about the story that we told you. And people remember details from the emotional story much more than the details about what they bought and where they were shopping and from the non-emotional story. Right? Then they redid this experiment. And before they gave people the emotional story, they gave them a norepinephrine blocker, a beta blocker, that's going to block the release of norepinephrine. And if we block the release of norepinephrine during the emotional component, they don't remember those components any better than they do here. So this is sort of showing that when people hear that story about the boy getting hit by the car and having to go to the emergency room, that norepinephrine is being released in our brain and is helping us remember that story more. By blocking that, they don't. All right. In addition to having norepinephrine released in our brain, we're releasing it in that fight or flight system. So when you get stressed or have an emotional event, we release norepinephrine. It causes overall alertness, increase of alertness in the brain. Sometimes we feel like we're thinking faster or processing information quicker during that time. It also increases sensory input. Again, all of these things would be very important for escaping a physical threat. The amygdala, again, becomes active, and there's a hardwired connection between the amygdala and the hippocampus. And when those things are both active at the same time, and there's negative events happening, particularly for prolonged periods of time, then some really negative things happen in the hippocampus. Right? And we'll talk about that in a little bit. All right. We're also, again, diverting glucose away from the brain and sending it to our muscles. Again, that 30-minute sort of stress response happens. And so we have an upregulation of blood pressure, upregulation of energy use in the body for 30 minutes. But if it's a psychological stressor and that's prolonged, then we start to run out of that glucose. We start to lose that sustained effect in our body. So we actually get the opposite effect for long-term stress. And in the short term, glucocorticoids are facilitating learning, they're facilitating memory formation, LTP, in the hippocampus. All right, so there's an inverse U function of stress and learning and memory. And so short term, we're going up, we're increasing memory, but long term, the U starts to come back down and we start to have decreases in function. So low stress, low to moderate stress increases function in the hippocampus, but severe or prolonged stress decreases function, right? And again, norepinephrine, as we saw with the Cahill study, is responsible for some of that memory formation. And in severe stress, the continual presence, though, of norepinephrine starts to interfere with LTP, and long-term presence of glucocorticoids can actually cause the opposite of long-term potentiation, can cause long-term depression, which is going to inhibit memory formation. So prolonged stress actually is counterproductive to memory formation. That's why you shouldn't be stressed when you study. You're making it harder to remember stuff. As attention is diverted from this to focus on the stressor, we also are negatively impacting the frontal lobes. So we have decreases in the ability to make good decisions, executive function, motivation, things go down, right? And the retrieval of memory starts to become compromised as well. So not just inhibiting formation of new memories, but being able to find old memories becomes problematic in the brain as well. We think that has something to do with the amygdala being hardwired to the hippocampus. All right. Sometimes we have the presence of things that are like glucocorticoids. Steroids act like glucocorticoids. And when people are on steroid anti-inflammatory medicine, so what condition did we talk about using that as a short-term treatment? MS, yeah. So in MS, sometimes people have memory disturbances because they're sort of artificially having high levels of glucocorticoids while they're on those. Non-steroidal anti-inflammatory drugs, NSAIDs, don't show that same effect, right? So clearly an effect of the glucocorticoids. If we do a research study and we just give people synthetic glucocorticoids, they start to show memory deficits within a few days. This looks like it is directly related to explicit memory, which is controlled by what part of the brain? Just take a guess. In your case studies, your video case studies you watch, do you have problems with explicit memory? And what part of this brain was damaged by the virus? The hippocampus, right? Yes, the hippocampus. Implicit memories. Remember HM couldn't remember things, but if you gave him a puzzle, what happened the next day? He was better at it. He had learned implicit. He didn't know he learned it. He didn't remember he'd never seen the puzzle before. Implicit memories, not through the hippocampus, probably through the cerebellum. Not affected by this. So here's experiment that was done with rats, and so they have rats that are in their home cage or in a chamber in which they have, well, some are in a chamber with just by the chamber itself, but then some are put in this test chamber and exposed to the smell of a cat, a predator, that initiates a stress response. And so we see cortisol go up in the rats that are exposed to that. And then we give them a spatial memory task, give them a radial arm maze or a Morse swim maze test. Those are particularly use the hippocampus, spatial memory, right? And the errors go way up in the animals that have released cortisol. The animals that didn't release cortisol don't have as many errors. Then, if we look at the excitability of hippocampal neurons, you can go in after this is done take a slice of their hippocampus and see how excitable the hippocampal neurons are, we see that in the high cortisol case, a lot of errors, hippocampus not functioning well, not excitable, hard to get it functioning. Compare that to the control animals, they have very excitable hippocampal, nice functioning hippocampus neurons. So now we need to talk about these two different things, right? So again, glucocorticoids are steroid hormones, and they bind to GR and MR receptors. But the MR receptors have a 10-fold higher affinity. What does that mean? What does it mean that they have a 10-fold higher affinity? What does affinity mean? Come on, Lee, you want to say it?
Not really.
No.
This just means they're more receptive to the.
Yeah, so affinity, we can talk about affinity like attraction, right? So they're 10 times more likely to bind to the GR, to the MR, sorry, than the GR. So what does that mean if we have a small stressor, a small release of cortisol? What receptors are going to be activated the most? MR, right? MR are going to be affected the most. That's triggering memory formation. It's the GR, the negative feedback loop, they have a lower affinity. Right? And so activation of MR and GR causes transcription to happen, causes learning to happen. Can be influenced by norepinephrine, the presence of other neurotransmitters as well. And in general, MR and GR sort of oppose each other. So low levels of stress activating MR. MR is kind of the good receptor. Increased memory, increase release of oxytocin and vasopressin, and all of these things happen based on MR receptors. Associated with enhanced attention and acquisition. GR has the negative effects. So a little bit of stress, right? Going to activate GR, we're going to shut things down, right? But when we have prolonged stress, then we are activating GR much longer and we start to get these negative effects, right? So low levels facilitates memory formation and turns off the system. Sustained and high levels impairs memory formation. And often we have something like a psychological stressor that's overriding that negative feedback loop. So even though we have high levels, the negative feedback loop can't do the thing that it's designed to do. Just realized that I don't have my watch or my phone, so someone's got to start having me on the clock here. So someone's got to watch the time. Where are we? 10 o'clock? All right. All right. Someone tell me when I have like 5 minutes left, and then I'll rush through the rest of the lecture. All right, so again, I've already talked about this, but MR, high affinity receptor, LTP, that's memory, right? Low affinity receptor only becomes really active, I mean it's used for this negative feedback loop, but it really only becomes active in the hippocampus and in the amygdala when there's high levels or sustained levels, right? And that's then going to have the negative effects that we see. Can actually cause atrophy of axons. It can cause cells to die in the hippocampus while prolonged activation of GR. So this weakens neural memories. We need more associative cues to retrieve that. Why would we have that system? I mean, why create this in the brain? Something that's going to kill neurons in the hippocampus. What's the natural reason for this system evolving and existing? To deal with what?
To relearn so that you can protect yourself better.
Yeah, to learn to protect yourself better from what? Short-term threats, right? Physical threats that are short-term, right? What if you're a zebra and you're constantly being hunted by this lion night and day for a week, right? The physical threat didn't go away. Right? Do you think that the zebra wants to remember all of that really horrible stuff? Probably not. So there is sort of a evolutionary protective benefit. If you're under prolonged severe stress, maybe you don't want to facilitate memory. Maybe you want to not remember all of this negative stuff that happened so it doesn't in the future cause problems. All right, so what happens neuroanatomically when we can see under high stress, we can see what we call transient atrophy. So transient means that it can come back, right? So we see a weakening of circuits, but we're not destroying those circuits. However, with long-term stress, we can actually destroy those circuits. We can actually have neurons become vulnerable to apoptosis, and we can lose neurons in the brain, in the hippocampus, and decrease the effectiveness of our ability to find previous memories prior to the stressor that have been stored, right? So almost like an amnesia effect. The memories aren't erased, they're just harder for our brain to find them under this prolonged stress. So here we see normal hippocampus, nice pyramidal neurons that have long axons that are going out into the temporal lobe to stimulate neural networks. Here we see, after prolonged stress, a decrease in the number and a disorganization of those hippocampal neurons in the hippocampus. So you can physically see those effects of prolonged stress. All right. If we lesion the amygdala, or we cut this connection, this hardwire connection between the amygdala and the hippocampus, we don't see these negative effects, right? And so we know the amygdala plays a role in suppressing memory and decreasing the effectiveness of the hippocampus. It has to be coupled with a negative emotional stressor. So people, remember I said that people that were on steroids for treatment of anti-inflammatory disorders will show some memory deficits. But even if they're on those for long periods of time, they don't show a loss of neurons and atrophy because we're artificially stimulating this and the amygdala is not active while those steroid drugs are present. So you have to have the negative emotional activity in the amygdala paired with the glucocorticoid activity in the hippocampus to cause these negative effects. Right? It's not seen in these other states, right? We have cortisol released in response to rewarding and positive stimuli as well, right? You can become aroused from positive stimuli, but we don't see these negative effects. Cushing's syndrome is a disease state where you abnormally release lots of glucocorticoids. And again, just the high presence of glucocorticoids not paired with amygdala activity don't show these negative effects. All right, so back to PTSD. So we know that in stressful events, right, norepinephrine goes up if you're in a crowded train versus not in a crowded train. If you have an exam, the night before the exam, norepinephrine goes up, right? We know that stressful events causes this. And if we block the autoreceptor, so the autoreceptor is a feedback mechanism, so if we block that feedback mechanism, it causes norepinephrine release, because locus coeruleus can't detect how much it's releasing. That induces symptoms in PTSD patients. One of the intriguing things, though, so I drew this on both sides because I want to show the difference here. So we have individual differences. So we can have one individual on this side, right, that has 50 GR receptors. So maybe that number, let's say 50, right? We have another individual over here that happens to create a lot more GR receptors, 200 instead of 50, right? And what we've noticed is, you have a platoon of soldiers, they all experience a traumatic event, but only some of those soldiers develop PTSD. Others do not. They all experience the same thing. Why are some people more susceptible to PTSD than other people? And a lot of research has been done in this. And what we find is that the people that tend to be susceptible to PTSD tend to have lower circulating cortisol in absence of a stressor. What controls the amount of cortisol that's just normally floating around in our body? What's regulating the cortisol system? Negative feedback, right? So if I have 200 receptors here, what is my circulating level of cortisol going to be? Is it going to be lower or higher than someone who only has 50 to control that? Does this one have a stronger or weaker negative feedback system? Stronger. So what does that mean about how much is being released here just normally? A lot lower, right? So lower baseline glucocorticoid. I'll take a picture of this. Lower baseline glucocorticoid is indicative of having more GR receptors, So now we have someone who has more GR receptors, less GR receptors. Big traumatic event happens, and we're releasing 1,000 cortisol molecules. Whose system is that going to impact more in the hippocampus? If we have 1,000 GC here and 1,000 GC here. This side, right? So activating 200 receptors full on versus only having 50 to activate full on is going to produce much more negative effects. And hence why we think these people are more susceptible to PTSD. So the idea is that the more GR receptors you have, the higher your sensitivity to PTSD. But in non-traumatic events, probably better able to cope with stress, better able to turn your stress system off, lower baseline circulating levels. So it's kind of this paradox, like these people are actually better copers with low amounts of stress, but in response to a traumatic event, their system is more susceptible to damage.
Is the amount that you have just based on your genetics?
Yeah. Genetics, oh, no. Yes, genetics, and one thing that we know is that through epigenetics, through environmental influences on our gene expression, particularly in the prenatal and postnatal phases of development very early in life, sets up our system here. So rats that receive lots of postnatal care have stronger systems, but also might be more susceptible to PTSD. Rats, if we prevent their postnatal care, they end up having overactive stress systems. Okay, treatment. Where are we on time? Someone help me. 12. Okay, let's do this. All right, treatment. You might need to medically treat some of the physical symptoms like sleep disturbances, arousal states. These people may need some anti-anxiety medications, something like that. Right? Often there's comorbid, we talked about comorbid things, right? Have anxiety present. We also may have depression because of dealing with this and feeling helpless or vulnerable. And then cognitive behavioral therapy is a primary therapeutic treatment. In particular, a subset of CBT is called exposure response prevention, or ERP, exposure response prevention. And it's a type of conditioning. So essentially what we think happens, and hopefully I'll show this on the last slide, But what we think happens is that the traumatic event happens, and there becomes this learned association between stimuli that are associated with this traumatic event, and those then trigger the anxiety and the panic attack associated with PTSD. And so by exposing people to those and preventing them from having a stress response, we can weaken that learned association. So it's like extinction in learning and memory conditioning, operant conditioning. Also, we're giving people coping mechanisms. So what are the appropriate coping mechanisms? Instead of turning to alcohol or drugs to self-medicate, we want to teach people other ways to deal with their stress. And there's a high comorbidity of addiction with PTSD because people often do self-medicate to try and alleviate their symptoms if they're not getting help. And high rates of suicidal ideation and suicide attempts in particularly like veterans that are self-medicating and not getting treatment. Changing our cognitive thought patterns, and then sometimes, we'll talk about this in our research papers, but blocking norepinephrine release during REM sleep can enhance the therapeutic treatment. All right, so here's an example. Let's pretend that you're having a friend coming over for dinner at 7, it's now 8 o'clock, they haven't shown up, and they haven't called you, you can't reach them on their cell phone, and they're not responding to your texts and your Snapchats and whatever else things you do, right, to kind of communicate with you. So how's that going to make you feel? How's that make you feel, Sydney? Sad?
A little stressed.
Worried? Anyone angry? Yes, Hayden's angry, right? Any other? Annoyed, right? All right, so we have cognitive thought patterns, right? Why do you feel sad, Sydney? What's the thought that's making you feel sad? Why they ditch on me, right? Okay, yeah. Feeling rejected, right? So you're thinking about being rejected, and that causes you to feel sad. You're annoyed because
They didn't tell me? Or
yeah, right? And then, what do you do then, right? Are you going to unfriend them on Snapchat? Because you're sad, right? I mean, there's some action, right, that you do, okay? So, you know, if you think they might have been hurt, maybe that makes you feel anxiety, right? If you know, right? Or you're anxious, you might get annoyed or angry if they don't let you know, right? But you could say, well, it doesn't matter if people are on time, right? That might be a different, when we talk about changing cognitive thought patterns, how do you interpret the fact that they're not showing up? It could be like, oh, I get an extra hour to like clean up more and prepare better, right? You can actually make it a positive thing, right? It's not our normal, probably, response to that, right? And so this then drives like what we do, right? And so this is the whole thing. If you're stuck in this mode with PTSD, right, we want to learn how to interpret those things in a more positive way and rechannel our energy in a more positive way. All right. So there's a bunch of different cognitive or behavioral outcomes that you can do. Some of them are not as positive, right, as others, right? And these are not independent from each other. So how you feel often directs your behavior, right? Okay. Where are we? We'll go over. We're getting close. I feel. 3 minutes. Okay. Let me pull all this up. Oops. Okay. Behavioral therapy is weakening the connections, the learned associations between the negative stimuli that we're trying to avoid, right? That's part of this, persistent avoidance of stimuli, right? And then the cognitive is helping us rethink how we interpret things, change our cognitive patterns. All right. These are the important things of the cognitive components. There's 4 important parts of that cognitive therapy. We're relabeling, right? These anxious thoughts are not real. I don't have to be afraid of the big truck coming towards me because I'm not still in Iraq and that's not going to have a bomb in it like it did then, right? Relabeling that these are not real, these are false signals reattributing it to the fact that it is, I have this problem in my brain and that's what's causing this. It's not psychological, like there's an actual physical, medical thing that we're treating here. Now I'm going to refocus my attention into more constructive relaxation techniques, ways to deal with the stress. And then after it's over, I'm going to reinforce all of that learning by revaluing my ability to do this, my ability to do this. So relabel, reattribute, refocus, and revalue. This consists of teaching the patient, like, what's going on in their head, right? Neuroeducation about what's going on. Weekly meetings and giving them assignments with exposure response prevention that they need to do throughout the week. Having them monitor their progress, creating a diary so that they can see that they're getting better. They can see their progress, right? This is the exposure response prevention. We're not going to do it like this, but we're going to do it in baby steps. We're going to work towards the end game, starting out slow. There are several different relaxation techniques that have shown success, very high success rate with this. Okay. All right, does it work? Yes. So this is a study that looked at mindfulness meditation. And what we see is that in the mindfulness, we can actually influence our brain activity compared to the controls. So practicing these techniques actually changes our ability to control our brain activity, changes our ability. And these are areas that are associated with pain, associated with learning, memory, self-awareness. All right, so this is the last slide. So what we think happens to PTSD, we get this single trial learning, exposed to a horrific event, this learning habits in our brain, and it takes ERP to disassociate that and cause extinction, right? However, even if we do extinction, sometimes after some period we can have spontaneous recovery. So this is why it's important to train our brain to think about things in a different way, to change our cognitive patterns. And it also shows the benefit of booster therapy for someone to come back in every 3 months, even if they're not having symptoms, to sort of reinforce what they learned in case they reemerge as a spontaneous activity. Cool? Good? All right. Read those 3 papers. We'll try and squeeze in some case studies as well on Wednesday. I'll take a picture and post this to the Moodle page. See you then.